My name is Wes Goar, and I am a senior bioinformatics scientist in Alex Wagner's lab at Nationwide Children's Hospital. Today, I am going to present my work on annotating VCFs using the GA4GH Variation Representation Specification, or VERS for short. First, what is VERS and why should you care about it? VERS is an extensible framework for the computable representation of variation. VERS provides semantically precise representation of variation. VERS is important because it enables the federated identification of biomolecular variation with globally consistent and unique computed identifiers. Simply put, by applying VERS to sequencing annotation, genomic knowledge bases, and electronic health records, we ensure the precise and reliable exchange of information regarding variation. VERS is an open source package and is freely available for community use. Please see the VERS paper if you would like to learn more or follow us on our GitHub. Let's briefly talk about the overall architecture that is required to use our VERS annotation script. Seq Repo and the Universal Transcript Archive, or UTA, are both databases found within the BioCommons package on GitHub. Seq Repo provides access to sequence data, while UTA provides us access to alignment data. These two resources are crucial for the normalization and translation of variants into their VERS representations through the VERS Python package. The VCF annotation script works by following this simple flowchart. First, we read in and parse the VCF by leveraging the PySam package. Then, we convert each VCF record into a nomad-style format to allow for the normalization and translation of both the reference and alternate alleles into their VERS representations through VERS Python. Once the VERS representation has been created, we add the VERS allele IDs to the info field of the VCF. Finally, we output a new VCF that has been annotated with the VERS allele IDs, and the user can also choose to output a pickle file with a full VERS object for each VCF record. To benchmark the overall speed of the VCF annotation script, we annotated the GR-CH38 genome in a bottle VCF. This VCF contains over 7.8 million alleles, which were annotated in a little over 98 minutes, averaging approximately 1,300 alleles per second. While this is a good first pass, in the future we hope to increase the speed and efficiency of the VCF annotation script. So let's now return back to the question of why this is useful. Once you have annotated your VCF with the VERS allele IDs, you are able to use those as precise and simplified lookup values for federated data from knowledge bases such as ClinVar and Civic, who are experimenting with adopting VERS. You can also perform your own VERS annotation of any locally stored databases to greatly simplify the knowledge base searches and their required translation for proper annotation within your own pipeline. The goal of VERS is to facilitate reliable exchange of genetic variation data to maximize the personal, public, research, and clinical value of genomic information. We hope that the adoption of VERS at large will allow for more robust annotation of knowledge surrounding genetic variation, thus improving both research efforts and clinical care of patients. If you would like to find out more about VERS or VERS Python, or even get involved and contribute, please scan the QR code on the slide and check out our links. This work wouldn't be possible without all of my wonderful lab mates and collaborators. I would also like to thank all of the involved organizations and my funding sources. Thank you. Have a great day.